Good afternoon, everybody, and welcome to the last seminar of this season. And I'm really happy to introduce our speaker, Katharina Ruthsatz. Katharina was telling me about her past, and I think it's quite interesting because originally she was training to become a teacher, but she chose biology as one of her major discipline, and in the end she chose science over education, although I think you're still involved quite a lot in science communication. And she did her PhD at the University of Hamburg and is now a junior group leader at the Technical University of Braunschweig. And with that, Katharina, the floor is yours.
Thank you very much. Welcome everybody. Thank you so much for having me here today. I'm super happy about the opportunity for giving the EBD seminar today, and I would like to start with quickly introducing myself. As already mentioned, I'm an assistant professor and junior research group leader at the Technical University of Braunschweig, and there I'm working in the division of evolutionary biology led by Miguel Vences, but I've recently established my own working group that is mainly addressing conservation physiology questions. And before I moved to my current institution, I obtained my PhD at the University of Hamburg, so in the northern part of Germany, and I did my PhD in biology and ecophysiology. I also did my first postdoc at the University of Hamburg, and then I moved to Braunschweig. Even it's quite obvious that I'm crazy about frogs and tadpoles in particular. My research interests encompass a broad range of aspects related to animal ecophysiology. And some of you might have seen already me around here at the EBD because I'm currently a visiting researcher here for 2 months in the Evo group. And what can I say? It's my 3rd time here, so it seems like I'm really liking it here. And today I would like to give you some insights into my recent work in which my group and I basically link life history of amphibians with their physiology to assess the vulnerability of amphibians to global change. At the moment, our Earth is burning, it's sweating, it's drying, and it's getting more and more toxic due to climate change, habitat degeneration and fragmentation, increasing agricultural use of landscapes, urbanization, environmental pollution, and the introduction of novel species and pathogens. And these drivers of global change expose wildlife to a wide array of chemical, physical, and biological environmental stressors. And these environmental stressors have been attributed to declines in species and population worldwide and are so responsible for the ongoing biodiversity loss in our natural systems. Today, more than 40,000 species are threatened with extinction, and considering the IUCN Red List, more than 14% of all bird populations, more than 26% of all mammalian populations, and more than 41% of all amphibian populations are declining right now and so are threatened with extinction. Based on the IUCN Red List, we can consider amphibians as particularly sensitive to environmental stresses. This is in part due to their low dispersal ability and their generally reduced capacity for active habitat choice. But it's also due to their sensitive and semi-permeable skin that is basically like a sponge for pathogens, but also for aquatic contaminants. And it's also related to their amphibious lifestyle because they are migrating between aquatic and terrestrial habitats. That is, of course, linked to their hormone-dependent metamorphosis that can easily be disrupted by, for example, endocrine disrupting chemicals. Amphibian population declines are often silent but steady, especially if amphibians are not directly exposed to, for example, habitat loss, which would be detrimental, but are exposed to environmental stresses that lead to sublethal effects on vital rates, such as growth, development, metabolism, and reproduction. But amphibians can avoid a fitness decline in the face of global change in 3 ways: by moving, by evolving, or by being plastic. However, the capacity for migration is often reduced due to life history, species-species interactions, and also habitat fragmentation. And genetic adaptations are considered to be inefficient due to the rapid speed of ongoing global change. And considering this rapid speed, we can state that behavioral responses would be the most flexible and fastest option for coping with environmental stresses, but also phenotypic plasticity is quite flexible and kind of immediate in considering the rapid speed of global change. However, if amphibians lack the capacity to make use of one of these coping strategies, these populations will decline and go extinct sooner or later. I'm sorry, this is not really working. So the capacity for phenotypic plasticity might differ between taxa, species, and also populations, and might also be related to the kind of stressor an animal is exposed to. But most importantly, in animals that undergo a dramatic life history, such as in amphibians, where we have this complex amphibian metamorphosis, also the different life stages might differ in their capacity for phenotypic plasticity. So imagine one of these life stages we can see here lacks the capacity to make use of phenotypic plasticity. This life stage might pose a life history bottleneck for this population. So it is of key importance to not only assess the life stage specific sensitivity to a given environmental stressor if we want to determine a population's resilience to global change, but it's also of major importance to assess the life stage specific capacity to use phenotypic plasticity. And by combining experimental, field, and meta-analytical approaches, my group and I basically investigate how different amphibian life stages respond to a variety of environmental stresses. And we are interested in which underlying mechanisms allow for these, for example, plastic responses to environmental stresses, but we are also interested in across life stage effects of phenotypic plasticity and being exposed to environmental stresses. Amphibians often breed in shallow and temporary habitats that are very likely to dry up or at least warm up, and so exposing larvae to an uncertain and unstable larval environment before a metamorphosis actually allows them to successfully leave their larval pond. So a very common response to unstable larval habitats in amphibian larvae is developmental plasticity. For example, in response to increasing desiccation stress or increasing temperature variation and increasing mean temperatures. So what we usually see in ectotherms with increasing developmental temperatures is a pattern following the temperature size rule, with animals developing at a higher developmental temperatures being at a given developmental stage, for example, the onset of metamorphosis, being younger because of an increased developmental rate, and likewise smaller because also the period for growing is likewise reduced. So imagine the environmental temperature surpasses a specific threshold that we can consider as stressful because it's affecting an animal's homeostasis. We will see, hopefully, something like temperature-induced developmental plasticity. And the underlying endocrine mechanism that might allow for a plastic response in developmental rate in response to altering or changing developmental temperatures is the activation of the hypothalamus-pituitary interrenal axis, so the neuroendocrine stress axis, which leads to an increased level of the glucocorticoid hormone corticosterone. And this, often called stress hormone, is also known to synergize with the major endocrine driver of amphibian metamorphosis, that are the thyroid hormones. And so both hormones indirectly and directly increase developmental rate and push metamorphosis forward. So the thermal reaction norm, or the degree of change in developmental rate with a given change of the environmental temperature, is also related to the responsiveness of this endocrine axis. And it has been hypothesized that the responsiveness of this endocrine axis is determined by local adaptation. So what we did in a meta-analytical approach is assessing whether temperature-induced developmental plasticity is linked to local adaptation and or phylogeny in amphibian larvae. by compiling empirical data, and this resulted in 150 estimates for the thermal reaction norm, or our so-called plasticity index. What we found is the following. Indeed, we saw a slight increase of the capacity for temperature-induced developmental plasticity with absolute latitude. But when we were comparing the latitudinal groups, so considering tropical populations, subtropical populations, and temperate ones, we couldn't find a significant difference between these groups. But of course, we have to note that these are very artificial. Then, looking at the bioclimatic variables that might shape the capacity for temperature-induced developmental plasticity, only one bioclimatic variable remained in our models after model selection, and that was the annual temperature range. But here we couldn't find a significant relationship between this bioclimatic variable and the capacity for developmental plasticity. Also, there was no phylogenetic signal on species level in terms of the capacity for temperature-induced developmental plasticity. However, looking at our data set, we have to say that most of the studies we have included into this data set used model organisms and were conducted on the northern hemisphere, and so definitely not in biodiversity hotspots. And so we can conclude that this data set here has a massive phylogenetic and geographic bias that might be true for many, many so-called global assessments of specific traits. So we could expect maybe a more general or more pronounced pattern in either way if we would have a more equally distributed data set on global scale. So we wanted to put our meta-analytical results into context, and we wanted to assess whether indeed corticosterone is somehow mediating temperature-induced developmental plasticity. So, for example, if we consider this as the underlying endocrine mechanism allowing for a plastic response in developmental rate towards different temperatures, we would also expect different levels of the stress hormone corticosterone at different developmental temperatures. And this also way before the onset of metamorphosis in order to allow for the pattern we find here. So what we needed here was a longitudinal approach, and we needed to measure corticosterone levels in early developmental stages of amphibians in order to assess whether corticosterone is mediating such a response. However, the common methods used for assessing corticosterone in amphibian larvae are invasive methods. So, for example, assessing corticosterone level in tissue or plasma. And so invasive methods are not allowing for a longitudinal approach if we want to have repeated measurements. So luckily, there is a non-invasive method for collecting corticosterone, which is waterborne corticosterone collection. And this is a method that is increasingly used for assessing corticosterone in amphibian larvae. But since this is a very new method, or kind of a new method, it has to be validated for every new approach it is used in. So we were interested in which developmental stage would be best to use for using this non-invasive method in order to have a decent illustration of the actual endogenous corticosterone level. So what we did was at 5 developmental stages, ranging from newly hatched tadpoles to juveniles after completing metamorphosis, we assessed waterborne corticosterone first and then took a tissue sample of the specimens in order to compare both collection methods. And the first question we wanted to answer was whether waterborne corticosterone is indeed illustrating the development of corticosterone throughout amphibian ontogeny. And it's a yes. So we can see that both collection methods, the waterborne method here and the tissue method here, reveal a consistent non-linear pattern for corticosterone throughout ontogeny. And this is good because this means that at least waterborne corticosterone is biologically meaningful samples at the different developmental stages. And what we found here was only a significant correlation at pro-metamorphic larvae, which are very well developed amphibian larvae, for example, having completely developed hind limbs and toes. They are approaching metamorphosis but not being there. But we couldn't find a significant correlation in very early developmental stages or very late developmental stages. And this might in part be due to, for example, a mismatch between skin morphology and endogenous corticosterone level. But we can conclude, especially for our approach, that these late-stage amphibian larvae might be the best ontogenetic stage in order to assess corticosterone using these non-invasive methods, and so allowing for repeated measurements. And this is what we did. We assessed at all developmental temperatures in these late-stage larvae corticosterone release rate using this non-invasive method. And what we found was the following. Indeed, we could see a significant increase of corticosterone release rates with increasing developmental temperature, which was pronounced here at the highest developmental temperatures used. Now we aimed for a proof of concept in terms of the temperature size rule. So what we would now expect following this pattern found for the corticosterone level is also somehow a reaction of developmental rate in response to these different developmental temperatures, resulting in kind of a pattern following temperature size rule. And so we looked at age and size at the onset of metamorphosis, and what we found is the following for age, body mass, and snout vent length. We found a significant decrease in age and size with developmental temperature. Also, having a closer look on the relationship between waterborne corticosterone at a late larval stage and age at the onset of metamorphosis, we can see a significant correlation of these both measures. So, providing some evidence that corticosterone indeed is kind of mediating this plastic response in developmental rate. However, even if a plastic response at one life stage might be adaptive, it might incur costs at later life stages. For example, carryover effects on locomotory performance, energy budgets, or for example, immune system functioning. So we were interested to see whether the plastic response in developmental rate we found here is persisting across the metamorphic boundary, and if we can see some carryover effects in the juvenile stage here. So we reared our tadpoles until the completion of metamorphosis and 3 more weeks from then on in order to test for any carryover effects on locomotory performance. But we also wanted to assess whether we can still find the pattern for size we found at the onset of metamorphosis, or whether there was kind of a growth compensation. And we also measured corticosterone level in order to investigate whether we can still the pattern found here at the late larval stage. For locomotory performance, we found the following. We assessed, corrected them for the different sizes, and what we found is indeed a kind of a carryover effect of developing at different developmental temperatures on the locomotory performance, because this was significantly poorer in juveniles that had developed at higher developmental temperatures. And this is of importance because locomotory performance is directly linked to terrestrial survival in juveniles, because we have a link to predator avoidance, foraging success, and general movement. Looking at the size at juvenile stage, what we found for body mass and snout vent length was that we don't have the pattern we found at the onset of metamorphosis anymore. And this means that we have kind of a growth compensation that happened somewhere at some point between the onset of metamorphosis and 3 weeks after completing metamorphosis. But here we have to note that this for sure wouldn't happen under natural conditions, because what we did was providing food ad libitum. But this usually never happens, that resources are available ad libitum in the natural environment, especially considering that these guys differed in their locomotory performance. And then, last but not least, looking at the corticosterone level, just to have an idea what it looked like again at the late larval stage. This is what we found in our late larvae, and this is what we found in juveniles. So the pattern found here for the increasing corticosterone level in late amphibian larvae with developmental temperature was not existing anymore at juvenile stage, which is good because elevated corticosterone levels have been linked to poorer immune system functioning and also poorer responsiveness in terms of acute stressors. So this is good. And this brings me to the next step, because if we want to encounter higher developmental temperatures, we also need different thermal tolerances or acclimation capacity throughout amphibian larval development. And so we were interested in seeing how the acclimation capacity changes throughout amphibian larval development. And what we did here was raising tadpoles at 5 different temperatures, and then at, I'm so sorry. So what we found, what we did was at 7 consecutive developmental stages, we assessed thermal tolerance and also acclimation capacity. And then what we found for acclimation capacity was the following. Indeed, we found a significant change in acclimation capacity for both thermal limits, so for Ct-min and Ct-max throughout larval development. And what we also can see is that the acclimation capacity for Ct-max was way higher than for the acclimation capacity in Ct-min, which absolutely makes sense because it's more likely for tadpoles to encounter more temperature variation at the maximum edge than at the minimum edge. Also, we found that acclimation capacity in this population of Rana temporaria was kind of low, with 55% acclimation capacity in Ct-max and 15% acclimation capacity in Ct-min. But the most interesting finding we had here was the low acclimation capacity in the very young larvae. And this means that these young larvae are most sensitive and most vulnerable to temperature extremes associated with climate change. And so these early developmental stages might pose a thermal bottleneck for this population of Rana temporaria. So considering that exhibiting phenotypic plasticity is energetically costly, especially during amphibian metamorphosis, that is an energetically demanding process, we can expect also some kind of energetic costs of being exposed to environmental stressors. And so we were interested in investigating whether environmental stress per se can affect energetics of amphibian metamorphosis. Amphibian metamorphosis is driven by the thyroid hormones, that is the major endocrine driver of amphibian metamorphosis, but some environmental stressors are known to affect thyroid hormone levels through endocrine disruption on this endocrine system, leading to altered levels of the thyroid hormone. And this is of key importance in amphibian larvae because any effect on the thyroid hormone level during amphibian metamorphosis will ultimately affect amphibian growth, development, and energy metabolism. But what is the connection between thyroid hormone level, endocrine disruption, energy metabolism, and energy budgets that can be used to fuel amphibian metamorphosis? So the thyroid hormone level, if this is altered, this will have an effect on the developmental, on the metabolic rate, because it is very well known that metabolic rate is positively correlated with thyroid hormone level. If we see an effect on metabolic rate, this will also affect the energetic maintenance costs, and so the capacity to build up internal energy storages that can be used to fuel amphibian metamorphosis. So we can expect that any alteration or disruption of the thyroid hormone system will ultimately affect the energetics of amphibian metamorphosis in the way of changing or affecting the duration of this developmental period, the energetic demands during it, and the energy allocation in terms of how much energy can be allocated to maintenance costs and developmental processes. So we wanted to investigate how the endocrine disruptive effect of environmental stress might impair energetics of amphibian metamorphosis. And to that end, we exposed tadpoles of the common frog to either L-thyroxine or sodium perchlorate in order to alter the thyroid hormone level in two ways. So L-thyroxine, as I already mentioned, is one of the natural thyroid hormones, leading to an increase of endogenous thyroid hormone levels. And on the other hand, we have sodium perchlorate, that is an environmentally relevant endocrine disruptor inhibiting thyroid hormone production and thus leading to a decreased level of thyroid hormones. And what we found is the following at the onset of metamorphosis. So we couldn't find any effect of altered thyroid hormone levels on the metabolic rate at the beginning of metamorphosis. But interestingly, we found an effect on the size of energy storages in the way that it was reduced for both altered levels of thyroid hormone levels, independent of which direction the thyroid hormone levels were altered to. But then, looking at the actual period of amphibian metamorphic climax, we found that altered thyroid hormone levels indeed affected the length or duration of this developmental period, with those tadpoles developing at the highest thyroid hormone level, revealing the shortest duration, and vice versa. And so it's not surprising that also in terms of the total energy used to fuel this developmental period, we can see the same pattern as we found for the duration. But then, considering the energetic maintenance costs throughout the entire period of this metamorphic climax, we found that those tadpoles developing at the highest thyroid hormone levels revealed the highest maintenance costs. And this means that they had the fewest amount of energy that could be allocated to developmental processes ongoing during amphibian metamorphic climax. And so we can conclude that endocrine disruption will can affect the energy budgets that can be used during amphibian metamorphic climax, but also that altered thyroid hormone levels impact the energetic efficiency of metamorphosis, because we can see this shift from covering maintenance costs and covering developmental costs dependent on the thyroid hormone level. Of course, endocrine disruption during larval stage might also affect juvenile performance through, for example, carryover effects. And this brings me to my first get out of this section, because what's next? So next would be, or the most interesting part next would be assessing what's actually happening to the juveniles, because juveniles are the most overlooked or neglected life stage in amphibian conservation science, because we actually don't really know what's happening to them once they emerge from their larval ponds. Of course, a huge amount of them simply dies, if not the majority, but we don't know yet what's actually shaping their terrestrial survival. And so it might be a good idea to dive a bit deeper into size effects, energy budgets, locomotory performance, and thermal physiology, and how everything works together in order to shape terrestrial survival. And this might also give us some insights into the role of juveniles into amphibian population dynamics. But looking at the costs of phenotypic plasticity and across life stage effects of phenotypic plasticity, it might also be interesting to have a look on what's actually happening to the adults. For example, during reproductive season, during breeding time, what if adults during breeding time are exposed to stressful environments? Might we see this effect also on the performance, for example, of the offspring, or maybe on the coping capacity of the offspring? So even if some high altitude or high latitude populations are very well adapted to harsh breeding conditions such as snow, cold spells, or even ice, climate change is also changing winter climate in many parts of the world. And with this winter climate change, what we can see is a phenological shift in many early spring breeding populations and organisms, including temperate amphibians. And this might lead to longer activity periods and thus higher energetic costs. And also we can expect kind of costs of parents being exposed to thermal stress in that way, that they might be exposed to cold stress, because also with winter climate change, cold spells in temperate areas are known to increase in their frequency. So we aimed to investigate how cold stress experienced by the parents during breeding time affects the offspring's performance and coping capacity. And to that end, we collected 3 amphilectin pairs of the common frog before a cold spell and after a cold spell in March 2021. And we had a decent cold spell of 5 days, and temperatures dropped down to -5 degrees during the night when the amphibians are active. We also wanted to determine whether parental cold stress might affect the stress resilience of the offspring. And what we did was exposing the embryos to 6 different nitrate concentrations throughout development. And then at 3 developmental stages, namely in embryos directly after hatching, in amphibian larvae, and in juveniles after completing metamorphosis, we assessed first hatching success and hatching size, effects of parental cold stress on growth and development, and how this might affect acclimation capacity in metabolic rate in response to short-term temperature increases. And what we found was the following for the first life stage investigated for the newly hatched tadpoles or embryos. In terms of the hatching success, we found our late cohort, so these guys whose parents had been exposed to the cold stress, to be actually more resilient to increased nitrate concentrations. And we also found them to have a lower mass at hatching, in contrast to this here probably reduced and increased resilience to nitrate pollution stress. Looking at the amphibian larvae is the following what we found for the onset of metamorphosis. We investigated whether parental cold stress affects age at the onset of metamorphosis and likewise mass at the onset of metamorphosis. And we found the tadpoles from the late cohort being older at the onset of metamorphosis, but likewise larger at the onset of metamorphosis, indicating that parental cold stress affected not only developmental rate, but likewise growth, because they had more time actually for growing. Then, last but not least, we assessed whether parental cold stress might affect the acclimation capacity and so thermal sensitivity in metabolic rate to increasing short-term temperatures. And we assessed that via calculating Q10. And what we found in metamorphs and also in juveniles after completing metamorphosis is that we have a way higher thermal sensitivity in the late cohorts, so for those tadpoles whose parents had been exposed to the cold stress. And also we found this pattern at the higher nitrate concentrations in both cohorts. Altogether, this means that phenological shifts can reduce the offspring ability to cope with stressful conditions if, because of a phenological shifts, parents are exposed to, for example, cold stress. Because here we found that breeding after cold stress or cold spell decreased the acclimation capacity in the offspring. So what's next? There is much more to be explored in terms of the underlying mechanisms mediating these parental stress effects we found here on offspring performance, health, and survival. So the next steps will be diving a bit deeper into epigenetic effects, also into maternal glucocorticoid hormone deposition. So for example, if there is a stressed mother, will this also increase the corticosterone level in the eggs, for example? And also if there's any effect on the energy budgets from the mother to the eggs to the larvae. And I would like to end this talk with a project that is very near and dear to me, because I think besides doing research and also besides supervising, training the future generations of researchers, biologists, zoologists in my case, is an important duty of ours. And during the pandemic, my colleagues and I found ourselves in the situation that we had to teach hands-on dissection classes online. And this is actually not possible following the definition of hands-on. So we aim to develop a resource that is not only addressing the everyday world of our students, which is basically social media, especially was social media during the ongoing pandemic, but also helps our students to dive deeper and learn animal anatomy and also comparative physiology, and somehow replace the courses that couldn't take place during the pandemic. So the project Anatomy Insights was born. And for me, this actually was a great excuse to get these things finally started, because I've always been absolutely passionate about teaching these basic zoology classes, and I have been doing so since my early masters, so more than 12 years now. And yeah, it's a great passion, and I was absolutely excited that we could start this project actually. And Anatomy Insights is an open access educational resource that we mainly distribute via our YouTube channel. And here we are dissecting our way through the tree of life. And we that are Mark Scherz, Miguel Vences, and myself. And so far we have already uploaded an entire season of long and short clips in which we are dissecting different animal groups and give some insights into different techniques for dissections, but also try to communicate evolutionary patterns and to put everything into an evolutionary context. And the benefits of this project have also been highlighted in a correspondence paper recently. And besides sharing everything on YouTube, we also use other social media platforms, such as Instagram. And here we provide information that goes beyond the information we provide in our videos on different animal groups. And we try to provide this information at least once a week. At the moment, we are taking a short summer break, but we will be back soon. The great thing actually about this project is that it connects us with different working groups with which we usually wouldn't get in contact to. For example, in 2021, we were collaborating with the Natural History Museum of Denmark and the University of Copenhagen and helped them while sampling tissues for a ringed seal population. And it was great because we could do our filming while these dissections and being part of this monitoring project. And this was, it was simply fascinating. Also recently, we had the chance to take part in an ongoing student course at the University of Hamburg. And here we filmed a turtle dissection, especially focusing upon the circulatory system, because we are already planning a second season for this Anatomy Insights project. And in this second season, we aim to impart an equal evolutionary understanding of vertebrate anatomy and physiology, because we are convinced that, especially for ecophysiologists, it is mandatory to have a basic knowledge of how different system works and why, for example, different taxonomic groups respond differently to global change effects in order to determine their sensitivity. And we were very lucky because for the second season, we got some help from some people of the University of Glasgow, because we recently organized a workshop there together with Sean Kiln and Mike Rutherford from the Hunterian Museum of Glasgow. And in this workshop, we tried to bring together this classical hands-on experience with the development of a digital resource. And this digital resource is more or less the videos we will produce on these animations you've just seen. And this workshop was basically structured in 3 work packages. We started with the hands-on dissections of the different vertebrate groups, and then asked the participants to prepare a presentation on their in situ experience, combining everything with drawings and also photos. And what we will do for producing our second season is taking the results of the presentation and building this digital resource, which are basically the videos for the second season. It was a great fun organizing this workshop, and we are very lucky that the company of biologists has funded this workshop, because so we were able to get all the equipment we needed. One last thing I would like to mention is no animal has been killed for all these videos, neither for the YouTube channel nor for this workshop. We always using roadkills or animals that have been died because of age, or animals that are bred for example, the food industry. Because one long-term goal of ours is to synthesize or draw attention to the massive use of specimens used in high school dissection classes and also like biology basic classes at university. And we think that by producing high quality videos as we do, we can reduce the use of these animal specimens in high schools and universities. And I'm absolutely curious to hear your questions regarding my talk, research, and maybe also this project. But first and foremost, I would like to say thank you for having me here today, and also for meeting all around at the EBD. It's a great pleasure, and special thanks to the Evo group for hosting me the third time, and also for supporting my research. It's a great pleasure. And we all know that science wouldn't be feasible without an international network of collaborators. So special thanks goes out to my group, who's always so passionate about all the experiments we're running. Same applies to Miguel Vences, who's always supporting our research. And of course, also to the German Research Foundation and the Company of Biologists for funding our research. Thank you very much.
Thank you, Katharina, for a very inspiring talk. And before opening the floor for question, I wanted to say that you're here until the end of August, so that's also...
End of July.
End of July, sorry. So that's also an open invitation for those that want to connect with you while you're still here. So are there any questions?
Also fine.
It was very clear.
Thanks, Katharina, for your interesting talk. Very informative, anyway. And well, I have several questions, more detailed. But for instance, in the last section that you have dealing with false spring perception by the physiological system of animals, in concrete with frogs, the question is whether the groups, experimental groups you selected, you think that they may have some difference, for instance, in the size of female, egg size, and so something
yes.
regarding the
yes.
maternal effect stuff.
Yes, no? that's a good point, and definitely we saw a pattern, because what we see for Rana temporaria is that usually the big males and the big females arrive first on the breeding ponds, and this was what we found like to be also in terms of the egg size and also in terms of the larvae, that they were generally of a larger size. Yes, so this might also affect the outcome of our project. Yes, so there was a pattern, and also in terms of their body condition. So the body condition of the early cohort was way higher than the body condition of the late cohort.
Well, probably my memory fails, but I think that people from Uppsala, Ansi, Alex Richter, and so publish something, or they
yes.
work on this effect on
yes.
the
yes.
lane and so. But I don't know if you can compare the results with yours, or you measure different.
I think they have also worked on the Rana temporaria. But the experimental setup differed a bit. I don't remember for sure the parameters they have assessed, but for sure also developmental rate. And I think they found some similar patterns, and they also suggested to dive a bit deeper into what's actually mediating these physiological effects we see here, because nobody has done that before. Yes, there is something from Uppsala.
Well, my last question is regarding the corticosterone that is mediated by temperature. and is affected also particularly to pre-metamorphic or metamorphic stage. The question is that metamorph used to be in the ponds when the pond is drying, so the temperature is ever higher than during larvae, of course, especially in temperate species population. So what's about this question with the season? And so I don't know whether if there is a predation or maybe something. Well, it's very.
How do you mean that?
It's a very light question anyway. No, it's
how do you mean that, like, late developmental stages are more likely to encounter warmer developmental temperatures, or how do you mean?
No, what I mean is that sorry, but not many people. The questions that corticosterone act when the animals are exposed to the higher temperatures during their life cycle. So I don't know whether it implies something that the animal are adapted to these conditions in the function.
Oh, for sure, there might be kind of an adaptation, because I think
stupid question, I think, but
no, there are no stupid questions actually. So I would say yes, this is kind of an adaptation, because this goes hand in hand with late developmental stage amphibians looking for, or like choosing, warmer level habitats in order to kind of push a little bit, like the last tiny bit of development that they are still missing for approaching metamorphosis to get out of that pond. And so yes, it might be adaptive to have then the corticosterone response in order to make this last jump possible, actually, yes. So.
Are more questions?
Hi, thanks for a very nice talk. It's a lot of results. I don't remember it all. It was very nice. I was wondering about the first part on the court. How do you know that, the effect on growth and age at metamorphosis? How do you know that they are, caused by the court? Like, you only have a correlation. So I was wondering if you would have the possibility of putting court in the water and simulating the same effect.
I heard that there are some groups doing that, right? Well, we didn't do that for many reasons, but of course this would also be a great way of controlling for that, sure. Because this is why I try to explain it with caution, that it might be one of the evidences that court could be involved in mediating that. Of course, this would be like the last piece in the puzzle that we would miss to make it a round thing. Yeah.
It's just that I find it really cool that you can measure the court in the water. So if you can also have like a very, easy way of, increasing their court level, that would be quite nice as a tool to understand the mechanisms. So
it is, but it definitely requires validation for every new approach. And also regarding the different temperatures, we have to be aware of that when temperature is warmer, also the skin, of course, is more permeable. And so, of course, just because of that physiological reason of the skin, we will see an increase in cord levels. So also in terms of the different developmental temperatures, this has to be validated, yes.
Thank you. One more. Yes?
Thank you, Katharina. Very, very interesting. So in plants, we also know that if you put some stress on plants, all the developmental rates advance, so everything goes faster, like in frogs. So despite the cost, so you can get smaller sizes, lower reproductive outputs. But in the end of the day, if you get a broader picture of what's going on, moving fast in nature, it's not bad. Basically because, you avoid problems only if you have a safe stage that can get longer in the wild. So do you think that this is the case, and despite in this climate change scenario we are in, there's some room from amphibians, to find this new niche in which you can move as fast as possible despite the costs, but increasing the safe niche to survive, to endure during hard times?
I would say yes, because otherwise, if they wouldn't survive like their larval stage, because, for example, ponds are drying out, we would see this bottleneck. So yes, it is highly adaptive, and it is absolutely needed for the amphibians to survive. And I think some people are always arguing that a small size at later life stage is of disadvantage, but I would in part disagree, because just because of being a smaller size, this doesn't mean that it's all bad, especially considering, for example, food availability. So if you're of a smaller size, you also need less energy, for example. So this could also pay off, like in later life stages, rushing through metamorphosis.
Yeah, but as I see it, there's a kind of limit, and that's the challenge, right? To find those limits. In Arabidopsis, for example, these fast cycles that we call, so you know, very long seed dormancy in the soil seed bank, but very fast flowering times. So small plants that just close that complete the life cycle in a short period of time. Surprisingly, what we find in those populations are local mutations, specific mutations in functional mutations in key genes of the life cycle. So it's not only plasticity that matters, but you need something else which requires time, you know, to become adapted to move fast in the wild. That's I think that the challenge, right? That can mask the pattern of what's what's going on.
That's true. Just focusing on plasticity might also simplify some things, but it's one aspect, but not the entire aspect. Yes, that's true.
Thank you.
Thank you.
Thank you. So we have two questions from Agus Camacho that's watching us online, but he starts by thanking you for your beautiful talk. So first question, Agus is wondering how many degrees this percentage is in acclimatation capacity, about 50%, of tadpoles translate to. Is it several degrees or below 1?
Can you please repeat it?
Yeah, I'll get closer. So he's wondering how many degrees these percentages in acclimatation capacity of tadpoles translate to. Is it several degrees or below 1?
That's a question. How does he mean?
I don't know.
What's like the slope of the thermoreaction norm, or what is he referring to? So maybe if he's still available online, please give us some details on your question.
I'll move to the second question, and Agus, if you can clarify the first, that's great. So let's see if we're luckier with the second. Which you think would be the rationale to apply this data and techniques to estimate the vulnerability of amphibian populations to climate change?
That's a pretty good question, because here we are just using an experimental setup. Everything is completely under control, and it's definitely not representing like the environmental temperatures they would encounter in the natural environments. So patterns might be a bit different in the natural environments, but this is like the first step, looking into if there's any capacity for acclimation capacity at all. So this first step solved, knowing that there is kind of acclimation capacity, could contribute in part to that programs or whatever, but I think, and this actually applies to many meta-analyses, that we should always be aware of, for example, these phylogenetic bias we found, geographic bias we found, and that we always have like a biased data set. So if we take the results from this biased data set and generalize it, for example, also for political actions, this could give us a hard time because we might overlook some important aspects. So yes, might contribute to specific actions, but we always should be careful with how to use it.
Thank you. Yeah, well, Agus typed, let's see if he's, I'll give him more time to finish if he has to add anything.
No, it's very. Thank you very much for your talk. It was very interesting. I'm interested about the meta-analysis results, and I was wondering whether you think that maybe, like, well, I didn't understand well whether you had like different species. How many species do you have? How did you like put the coordinates of the species in the map? Whether like you got natural populations. And
area for including data. We only took data from experimental setups, like from laboratory studies, because this is the only thing that can be conducted under controlled conditions. So we didn't include mesocosm experiments, and we didn't include field experiments. We also included only studies that used more than 2, or at least used 2 temperatures. And speaking for species, I think we had like 70 species included, like caudates and endurance. Yes.
And all like taken from natural populations and kind of endemic from different places?
Yes and no. Yes and no. So some of these were taken from natural populations, like the majority, and of course some were taken from artificial breeding, like hospital breeding or university breeding. These ones were excluded from the latitudinal analysis because we can't consider any local adaptation, especially, for example, model organisms that has been reared under artificial conditions for generations. Maybe they might lose phenotypic plasticity to some extent. This can happen, so we excluded them for this analysis, yes.
Yeah, okay, so great work about that. But like, do you think like, what was the missing point? Do you think that with an increased number of species, you might be able like to find like that pattern, or like was it like why do you, or like it was rather this lack of places from the tropics?
Well, it could be either way, but it would give us a more clear pattern, if there's a pattern at all or not. I mean, also finding no pattern is a result, and would be a good result, because then we would know, okay, the capacity for temperature-induced developmental plasticity is not associated with local adaptation. But the more data we would have in our data set included, the more trustworthy would the data set be. And it's only representative if we would include like an equally distributed data from all different regions. Yeah.
Thank you.
Thanks. So I have a reformulated question from Agus. So CT min and CT max change during acclimatation treatments.
Yes.
And your graph showed about 50% in acclimatation capacity. So how many degrees did CT min and CT max change? Thank you.
I tossed the slide actually because it was too long. So this is how CT min and CT max developed throughout ontogeny, and think here we can see that CT min didn't change so much at all. It's just a slight decrease to 40 degrees, and then we see a small increase in CT min, but it was like between 5 degrees and 7 degrees, in contrast to CT max, where we had a very low CT max of like 26-ish, and we got out by, I think, 36 or something. So this is a way larger range, which actually makes sense ecologically spoken, because as I already mentioned, they are more likely to encounter like temperature variation and temperature extremes at the maximum edge than at the minimum edge, because minimum edge would at some point be freezing, and then we wouldn't see any survival at all. So hope this answers the question.
Thank you very much. Are there more questions in the room? Yep.
Yeah, congratulations for your talk. Just a small point for your phenological shift, because you said that one problem was that maybe the advanced phenology could later take like frost events or cold events, no? And I think this is a temperate bias, maybe you have in your mind. So just for the proposal, something you need to write, that here maybe in the Mediterranean area is not so important. I mean, obviously you can have a frost event maybe in March or in April, but I think that maybe here the challenge is the extreme heat events here that we have in southern latitudes. So to make a more general framework for the phenological shift that could be working both in the northern latitudes and southern latitudes.
That's true. We considered that for our experiment here because what we see for our populations is that they indeed start breeding earlier and earlier, and that we from time to time see this cold spells. So we were interested in whether these can actually withstand these cold spells if they encounter cold spells. And in another experiment, we also assessed freeze tolerance because this is like relevant like in the northern part of Germany. But of course, yes, you're right that this doesn't apply like for example, entire Europe, and of course doesn't apply to subtropical areas. Yes. Thank you.
Thank you. Any pressing question? No? So I think we can thank you again.
Thank you.
For this talk. This closes, well, you deserve a round of applause. And this closes this season's seminar at EBD. I think I can speak for Bia and myself saying it's been a pleasure to organize them. And so we'll see you on September 7th with Paula's talk with the new organizing team with Javier and Maria. And I don't know, is there a discussion group next week? So don't miss the discussion group next week. Other than that, see you in September then. Thank you. Thank you.